everyone, I'm Rylee. So glad to be here today and thank you all for coming. Today we're going to be talking a little bit about conservation genetics in the context of some rapidly changing conditions that we're experiencing. So my intention today is to walk through my experience surrounding this topic a little bit like walking through a story and taking you along the journey with me. I'm not here today to be presenting some really neat solutions. I have more questions than I do have answers, but I think that's what this collective is all about, because we all have such unique situations that we're working with. And I'll be talking a lot about Kingston today, but just to let you know, I'm not here representing Kingston, just here in my own capacity to try and contribute something hopefully of meaning to the collective. So what we're doing, getting together, sharing our experience, our insight, it only increases our capacity to navigate complex topics like this, which is so important. So a big thanks to Knox Environment Society for hosting, really appreciate it. So just before I begin, I'd like to acknowledge the traditional custodians of the lands in which we come together today, the Wurundjeri Woiwurrung people of the Kulin Nation. This is one of my favourite photos I've ever taken. It's one of my favourite places in the world, Bullock-Willum. You may know it well. It was taken back in 2020 while the world was in a bit of chaos. But yeah, these places are so special, scattered around, offering us a lot of respite from the world around us. And so I pay my respects to the Wurundjeri people who have cared for this country over millennia and continue to today. I pay my respects to elders past and present. So starting off, my background is in environmental science, terrestrial ecology, restoration, and I've spent a lot of time in the past, around 8 years in the field, studying plant ecology, learning about Australian flora, learning about threats such as invasive species, and conducting a little bit of research. Up the top there is just Anglesea, where Deakin spends a lot of time doing vegetation surveys. And then there's some lab work that I did in the lab at Deakin, though they aren't my slides. They're slides taken with a electron scanner. And then down the bottom there is some Langwarrin Flora and Fauna Reserve, where I did my placement removing Disa bracteata, and South African weed orchid. So just a little bit about where I started. And after uni, I spent some time on my own small business, which was a market garden farm, and that experience opened up an opportunity to start working as a grower at a place called Ball Horticulture. So Ball's a huge multinational commercial operation. Their facility down in Skye, you may have seen it, just off Hall Road. It spans over 30,000 square metres, produces around 90 million plants a year. And you can kind of see little yellow and orange dots in the back of that photo on the left there. Those are people. So the scale is pretty mind-boggling. I worked across mainly the germination, vegetative cutting and tissue culture greenhouses. And so within the big rectangles you see here, there's the kind of horizontal squares, rectangles. So they're different zones, growing zones, which can be set up for different temperature, humidity, sunlight irrigation, chemical scheduling, that kind of thing. So they have all the bells and whistles, refrigerated germination chambers, pneumatic bench movement, smartphone controls, all of that kind of thing. Lots of moving parts. So hundreds of different production lines, so many different teams. The timelines are very tight, 365 days of the year, Christmas, all of that. And the margin for error is really thin. But in saying all that, when I look back at my camera roll from the time, I can only find a handful of images of healthy plants, which kind of is indicative of the focus that I had as a grower there. Yeah. So pests, pathogens, chemical interactions, diseases, genetic problems, botrytis, damping off, you name it, it's all there, it's all still there, no matter all the technology and money you throw at things. And one of the biggest things I learnt during this experience was that, so yeah, it doesn't matter how much technology and money you have, the problems are much the same. The only thing that stands between failure and success with plants at the end of the day are people. So it keeps going, it keeps getting bigger. Teamwork, being determined to constantly problem solve, and having an eye for detail is still make or break. So I spent a lot of time in this space, developing that mindset, building my skills in growing and horticulture, and then I saw a position come up with the City of Kingston. And I was lucky enough to get it. So that kind of brings us to where I am today. So just kind of speed run through the past. So I work as a conservation officer with the City of Kingston, and I manage their Indigenous nursery there. So it produces around 30,000 plants a year, which is rather small, but it's focused on the bushland and foreshore conservation teams, and also departments like environmental planning, gardens for wildlife, that kind of thing. And if you've never heard of it, you're in the majority, because it flies under the radar. And I think mostly what I've gathered is because it doesn't have a retail component, which is very unique in the scheme of things. So it's funded entirely from internal Kingston funding. And so it's been set up so that the conservation teams at Kingston, they'll make a yearly to bi-yearly plant order, and they make that for their reserves that they're managing. So it's bushland or foreshore reserves, and usually anything that's left over is what the other departments get. So horticulture, gardens for wildlife, and these plants count towards various projects and targets that are under different council strategies and plans. So biodiversity strategy, green wedge plan, their well-being, council and well-being plan, that kind of thing. It all comes in some various points and parts within that. I'm the sole employee of the nursery, and I have around 6 volunteers that help me out once a fortnight. So it's very much still driven by volunteers. And before me, there was some very awesome women doing the exact same thing. You may know some of them, so shout out to them, Rafa. But when I started, compared to working at Ball, this is pretty much the complete opposite. The focus isn't about money, it's not about growing a business, it's about ecological restoration, protecting and restoring biodiversity across the municipality. There's no catalogue to buy seed from, there's no tissue culture coming in from overseas, there's no mother stock facility pumping out thousands of cuttings a day. It's just me and some volunteers. And so began a huge learning curve again. And I had a lot of questions starting to roll in, like where should I be sourcing plant material from? Where am I allowed to? Where has material typically been sourced from before, and what's the status of the flora populations? How are they travelling? What's their health like? And further, how fragmented are the reserves? How intact is gene flow? And really importantly, what ideas of provenance are dominant? So this is kind of where the start of the journey that I'm going to kind of take you on today. And again, don't expect a neat resolution at the end, but that's kind of the point of this collective, is that we, or at least what I see the value of this collective, is that we're facing all of these challenges within our different kind of spaces, and then let's bring them together and try and work them out. So begins the start of my investigations into those questions, and I'll just share some figures and stats, all of which are available in the publicly available biodiversity strategy and also the EcoFutures report that was made to inform this strategy. So within the City of Kingston, the overall protected council reserves make up less than 1%. And you can see these little blue dots on the screen are representing the spread of areas there. And historical records starting from around the 80s with flora surveys and ecological consultant kind of reports, they estimate that around 744 Indigenous species once grew across the reserves, and today around 440 remain. So meaning that around 40% of Kingston's Indigenous plant species have become locally extinct, which is very concerning. They estimate that only 9% of flora species found in Kingston are considered secure. So another aspect is that apart from the continuous strip of foreshore land, so you can't quite see it, it's very thin, but it just runs along that whole coastline there. That's all foreshore managed reserve pretty much. The largest inland reserve is the Grange, right up the top there, and that's around 6.5 hectares. So the median size of our reserves is around 0.8 hectares. So working with some really small areas here, and they're quite fragmented. So next part of my kind of investigation is to look into provenance, and what I gathered is that the prevalence strategy and practice is one that's been pretty common over the past, forever, is that material from, typically material should be harvested from the reserves and put back into the reserves, the same reserve. That's often kind of termed local provenance. You're probably all very familiar with that. But considering all the information that I just mentioned, it naturally started begging the question that if revegetation has been done in this way for the past 25 years, because that's when the nursery roughly started, what does that mean for the long-term outcomes of these populations, considering all these new effects of fragmentation? And for me, in my new role, how can I make sure that I'm utilising the best practices that I can when it comes to collecting seed and cuttings? So the main concerns that start to float around my head are to do with inbreeding depression. So that's what's happening when, well, it could potentially be what's happening when populations become small or separated. So the available gene pool is shrinking, and it's resulting in those closely related individuals to start to breed, which can affect the long-term viability of the population. So when we're thinking timescales beyond our own. So inbreeding, the term inbreeding can bring a lot of things to mind. The main thing that I focused on here is that when gene pools are shrinking, it increases the chances of two things, kind of similar but just different ways of thinking about it, is that increases the chance of harmful genes being expressed and also increases the chance of adaptive genes being lost. So kind of two sides of the same coin. Genetic drift is a concept that can amplify this. So it happens naturally and randomly, but when change in populations is accelerated, such as the rapid reduction in size, when things are getting smaller, more fragmented, genetic drift can have a big impact on the viability of the population, like what I said before. And I've got an illustration here that I found, which it does work, but you just kind of got to imagine the frogs as plants. You can see in the bottom row, there's a large population of frogs with a reasonably even mix of green and red frogs. And that's representing allele expressions. And you can think of an allele, this is a very overly simplified way, but you can think of an allele as a characteristic of an organism. So does it grow big? Does it grow small? Does it grow wide, or does it grow leggy? but also things like, does it tolerate heat, does it tolerate frost, and so on. So as they cycle through the generations, 1st, 5th, 10th, meaning they're growing, they're producing seed, and then the seed is starting the next generation, the large population is maintaining a balance. So the green and the red alleles are reasonably balanced. And they're pretty well positioned to survive through different effects. So again, thinking hot and cold, if it's tall, if a red one grows really tall, it's at risk of something like a windstorm or that kind of thing. Or if the green ones are really small, they're at risk of getting grazed or trampled, that kind of thing. But over time, those effects can kind of balance out in a large population. And then when we look at the smaller population at the top, we can see for whatever reason there's been a rapid reduction in the population. And so thinking back to Kingston, it could be fragmentation, it could be land clearing, that kind of thing. So as the generations progress, at some point genetic drift has maintained the traits of the red frogs. And then by the 10th generation, the green frogs aren't looking so good. It's almost taken out all of them. So this leaves this population quite vulnerable to future changes. So remember, theoretically, I was saying before, the red ones grow really tall and the green ones grow really short. And so if there's an influx of rabbits, for example, then the green ones are going to get knocked out. So that's just a kind of simple way of kind of viewing that. And then another concept that starts to become a concern is the occurrence of bottlenecking events. So these are quite major events, such as fire, drought, flood, major frosts, snows, and also disease. So myrtle rust, for example, or if you wanted to think of animals, the chytrid fungus in frogs. So survivors that emerge out of bottlenecking events can represent a significantly altered spread of genetic representation, of that allele representation. So again, creating vulnerability when they face future challenges. So you can see there's a nice spread of different colours in the original population. The event occurs, there's a surviving population that begins to take off again, and the orange has been significantly reduced. So it's the orange in this scenario still has a chance of surviving, and it just needs that time to recover. But another aspect of bottlenecking is when events start to compound. So you'll see this in a lot of threatened species research. For example, maybe you've heard of the Royal Botanic Gardens with their Banksia canii research. And so when we have those bushfires that are starting to happen more frequently, it's compounding that bottlenecking effect where we'll have a new generation of seeds start to grow, and then they don't have a chance to come to reproductive age and they get knocked out again. So reducing those, in this example, those orange alleles. So yep, that's all kind of coming up for me, and I'm thinking about these things. So, and then the last question, if we think back, was around gathering a sense of the different provenance perspectives. And I started to understand this from both the conservation team, ecological consultants, volunteers, the public, historical and current literature and reports. So it's all kind of developing from different areas. And the first and most prevalent idea is that remnant populations are optimally adapted to their sites in perpetuity, and so that crossing with outside genetics can result in an overall reduction in population fitness. So on the one hand, this is a valid and scientifically supported idea, which is often called outbreeding depression. So for inbreeding depression, and this is kind of starting to feed into outbreeding depression. And that's when breeding between distant relatives can disrupt local population traits or mix mismatched chromosomes. So mixing mismatched chromosomes we're going to get to in a little bit. But outbreeding depression can typically lead to the original population starting to lose vigour and starting to possibly affect their fecundity. So meaning that there possibly is a reduction in their reproductive success over time, meaning that introduced genetics and the original genetics are declining. So, but on the other hand, there's some subjective aspects at play here. The first is the perpetuity part, and the second is about the extent of localness. So I added some questions to my pile. It's growing rather large at this point, but it's still growing. So where do the boundaries of what is local actually lie? And what's the distance where populations are distinct from one another? And another important thing, what time frame does separation between populations typically start to create this divergence? And another one, and it's a little bit more hairier, I'm not going to lie. It's a little bit more, I suppose, sociological, but it's prevalent nonetheless, which is that the introduction of external genetics introduces inferior plants that will act to pollute the site history and integrity. So related to the last idea, but a pretty major language shift. And in a kind of simple way, it's kind of like looking at the reserves as a time capsule. So it comes up in reserves that have high quality attributes that are seen as representative of a type of environmental heritage, which is obviously cherished for good reason. But it does raise the question as to whether imposing these values acts to conserve the ecosystem in the long term, or is it just maintaining the appearance of one? Thinking about, functionality over kind of imposition of what we're thinking is happening here. And with all that, those provenance perspectives, it's not just about gathering a sense of what people are kind of viewing the provenance like. It's also about the implications. So the implications that these perspectives can have on management. And an example here is that in one of Kingston's management plans, conservation plans, is that there is an action that has been written in that's really dominant, that is to minimise or cease entirely the act of planting in high value reserves and rely only on natural recruitment to obtain new plants and maintain plant populations, which is tricky. It's tricky, it's not incorrect, it's just that we need to, I'm getting a sense of how do I make sure that these past decisions have been formulated under a particular set of conditions are still applicable today? Are they still aligned? And yeah, it's not about rushing to make decisions or judgments or anything like that. But thankfully, one of the first things that you'll come across when you start to look into these questions is Flora Bank guidelines. So you guys heard of the Flora Bank guidelines? Would have been a bit worried if no one had. I'd be in the wrong place. But a quick recap for anyone that may not know is that Flora Bank guidelines were introduced in 1999 when revegetation was starting to take off in the mainstream. 1999 had just concluded the decade of land care and also Bob Hawke's 1 billion trees program. So it's estimated that around 700 million trees were planted by 1996. So just to show the kind of scale of what was occurring at the time and what nurseries were kind of experiencing. So also in the late 1990s, we have the Kyoto Protocol start to come into play. And I mention this because that's kind of the emergence of offsets, carbon credits, emissions trading. That's all starting to arise, and it's resulting in these new demands from interests such as mining. So wanting plants for revegetation-based offsets all across the country, which we still see very prevalent today. And so it worked to provide some structure, some guidance, and also had a suggested code of practice helping to promote sustainable operations, ethical seed harvesting, collection, that kind of thing. And now I'm just going to jump around in time a little bit now just to kind of build this out a little bit more. So this is a very blurry image, but I'm kind of putting this in because as much as the City of Kingston is a good case study, it's not unique by any means. So when we look across the country over the past 20 years, anywhere we'll see some changes, some pretty major changes. So this is, we've got Cranbourne Gardens in the middle there in 1985. And this is just, drop a pin on Google Maps or Google Earth rather, and you can use they have got like a time scale that you can jump around in their images. And then this is 2025. So I'll just go back again one more time. And so Australia's become one of the world's 11 deforestation hotspots. We've cleared over 10 million hectares of vegetation since 2000. Yep.
Was that farmland or actual wild just land?
So it's a good question. It's mostly farmland, but there is still some really important grassland adjacent species that were existing. For example, if you imagine Pakenham area, Officer, that kind of thing, I know of projects that have taken seed from Pakenham and they've put it into a development in Kingston, and then the seed gets cleared from Pakenham from development. So all this stuff is moving around, and on the surface it does look like just farmland, but there is some still really important populations existing in these areas before development. Does that kind of... Okay. So another thing is that in Victoria we have the highest human population density of any Australian state or territory, which is kind of surprising. Historically also we've had the greatest proportion of land clearing and habitat fragmentation. So from 1999, the original Flora Bank guidelines, and then with all of that kind of context in mind, in 2016, 2017, Australian Network for Plant Conservation conducted the Australian Native Seed Survey. Did anyone participate in this at all? Yeah, it's interesting. It apparently went out to a very, very large kind of cohort of users in the kind of native seed sector. So it had the aim of better understanding current practices and getting some feedback on the challenges being faced. And it was trying to apply a modern perspective to the changes in the landscape and gauge a sense of the sector's capacity for meeting this ever-escalating demand for seed for native plants as it grows in complexity as well. So, and the main respondents, according to the report, were seed collectors, growers of seed production areas and seed purchasers, so within Indigenous nurseries as well as seed production farms. And the responses are pretty consistent across the board. So they found that there is a increasing pressure on wild population, which is really a main component of the report, but also that market for native plants and seed does not typically recognise the true cost, which you may be familiar with. It's that, demand is increasing, complexity is increasing, but we still consider the materials that come from wild populations as free and existing in perpetuity. So yeah, increasing demand for more species, general lack of seed available for that for these now more complex orders, and also despite all that, market demand is inconsistent, so it's resulting in some financial and staffing insecurity across the board. And this, so that survey was really integral to forming a full revision of the Flora Bank guidelines in 2020. So it was released in 2021, and there are 15 modules that they created covering native seeds and their supply chain, and a really high proportion of contributing authors and also was peer reviewed by 80 more. And it was trying to draw on 20 years of advancements in native seed research and understanding. So all of the modules are worth reading if you haven't read them before. I think they give a really good overview, and they have a lot of really good people that have been working in the field for a long time. One of them in the area that we're going to be talking about today is Linda Broadhurst. So yeah, definitely give it a look if you haven't seen it before. But the one I needed the most was number 5, seed sourcing. So I'm thinking, okay, case open and closed. I've got all the information I need, but not quite. So seed sourcing module happens to have some very helpful information when considering the provenancing strategy to use. And if I'm working within the dominant strategy in Kingston, local provenancing, it has some good information on what I should be trying to do when I'm trying to meet those minimum numbers in the field. But when I tried to meet the minimum numbers, I started to come into some trouble. And also, just to put in a little point that I'm not talking about threatened or endangered species here, I'm just talking about normal non-listed species. And yeah, this doesn't cover threatened species or endangered species. But it's trying to prevent those species from becoming threatened and endangered overall. And so there's 3 things, yeah, that I found more or less in tandem. So for a lot of species, when I tried to use this strategy, I couldn't get high enough population numbers to meet the minimum requirements. Another thing, I quickly realised that I was often directed to harvest from what was historical revegetation sites. So that's mainly a concern for me for cuttings, where I risk harvesting cuttings from plants that were previously cuttings from cuttings, so clones of clones of clones. And lastly, and kind of most importantly for some of those management actions that have been really strong for a long time, is that natural recruitment is not actually a given in the reserves and is actually starting to decline or not be present for a lot of species. So yeah, on to the next option, which is some of these other provenancing strategies. And the other provenancing strategies can sometimes, when it's this interesting kind of thing that I've experienced, where if I jump from local to something else, the response that I get is that, oh, you're going to start planting Queensland bottle trees in our reserves or something like that, where it's not, we're just looking at the spread of a population across the landscape and seeing whether with distance and time, considering distance and time, whether we can start to assist that gene flow again. So I'm going to just jump into the next one, but please do give this a read if this interests you. So the next step is planning to use another provenancing strategy. Planning being the key word. The guidelines answered a bit of my question at the very start regarding the extent of populations. So they highlight that, across the past 20 years, genetic testing has shown that what we previously considered to be a local population can actually span out much further than we had thought. For example, if we just take the top Eucalyptus ovata and we project that distance into Google Maps at 27 kilometres, we'll see that what could theoretically be considered a local population could span from here in Kes up the top all the way down to Rowan's Woodland in Kingston. Or if we wanted to go even further, the full 27 kilometres, we could go from Rowan's all the way up into Sylvan Reservoir. So it's a nice thought experiment, and obviously it's not about jumping into that kind of thing, but it's interesting that it's starting to break down those physical and imposed boundaries that split up the landscape in different ways. For example, this range here crosses Kingston, Dandenong, Knox and Yarra Ranges Council boundaries. So those are all imposed on the landscape. You know, the plants don't recognise them, but yeah, we do. So great news for me that research is starting to emerge. I'm thinking it might help with my dilemma. But 27 kilometres is pushing it a bit. So I'm thinking, how about just across the road? So I picked these reserves as a start just to test the waters and thought that surely this will be easy. It's kind of famous last words. So here's Kingston Council Rowan Woodland Reserve on the left and Kumura Nature Reserve on the right. That's Kingston Council, that's Dandenong City Council with Springvale Road splitting them in the middle. And it's where I encountered my first challenge, but it's just a minor one. And that's that my permit for plant material collection only allows me to harvest material on council land in the City of Kingston. And other permits, obviously, including the permits that maybe some of you have, go much beyond this, but because there's never been a perceived need in Kingston to get more coverage, that was what I was working with. And obviously we can update permits and that kind of thing, but I can't even find an email address for DECA nowadays. So anyway, that's another thing. The good thing is that Kingston has a really good relationship with Dandenong and even shares some tied conservation funding together. So that solves that problem. Either I could be granted permission under their permit, or we could do some sort of exchange and do that kind of approach. So that solves that problem. But the real roadblock, and I'm bringing back this slide from before, is realising just how deep some of the perspectives go. So remembering the example of outbreeding depression informing the idea of home site advantage. Next slide. And the environmental heritage perspective, and how both had been written into the management actions in the form of stopping planting and relying on natural recruitment. So I came into resistance pretty quickly, not because of permit limitations or anything like that, but because there's something here that's been operating for decades, and it makes sense when you look at it like that, that some fancy new guidelines, some, you know, vague, it's kind of vague to, we're not trying to transfer what the Eucalyptus ovata information is to another species because we can't. So they're not quite convincing enough to make the decision. So I have to keep digging. And actually what I found makes a lot of sense as to why there's still this kind of ongoing hesitance. And so the next level is the literature. I hope this is all making sense. It's a little bit of a rabbit hole, but bear with me. And a name you'll come into, a name you'll find very quickly with these kind of questions, because it's attached to almost every major paper related to Australian flora population genetics, is Richard Frankham. So this 2011 paper that Frankham and team put together a decision tree for this exact scenario. How do we mitigate the risk when introducing assisted gene flow as a management action? And it's about predicting the probability of outbreeding depression. This is a simplified version that I made, and you may notice that step 2 is missing, so we'll get to that. Number 1 is that, are you certain that they are the same species? Sounds obvious, but it gives particular significance to the potential of undiscovered species, subspecies and hybrids. For example, it's really important in species like eucalyptus, where you've got a lot of hybridization that could be occurring, and also Irididosperma, the wallaby grasses, where they can have some very specific within the species, but just slight variations. So that kind of addresses that. But for the purposes of my kind of investigation, let's say that we know for sure that we're going to take a cutting from the exact same species in Kimura, we're going to grow it and we're going to plant it in Rowans. So yes, we're going to move to number 3. Have these populations exchanged genes in the past 500 years? Yes. Springvale Road has only been there since the 1850s, I think. The bypass was put in the late 90s. Yes, they've definitely been connected, and accounting for intact pollination and all that kind of thing, they would have exchanged genes in the past 500 years. Number 4, is there a substantial difference in the environments between the populations? So we're going just across the road, but we will make sure that we're matching the EVCs, the ecological vegetation classes. We're going to check for any major differences like geology or hydrology, that kind of thing. And so we're going to say no, which tells us we can pass go, we can re-establish gene flow. And number 5, if we needed it, is again, it's just highlighting that dependence on the uniqueness of the species that you're trying to kind of assess. So there's species with very quick generational succession to consider if you were to go to that level. But then we have the missing number 2. Do they have major genetic barriers, otherwise known as fixed chromosomal differences? So initially when I read this, I could not for the life of me understand what it meant. I thought, here's a decision tree with all the other steps appearing reasonable, easy to find answers for. You can take a really cautious but still effective risk management approach, and you can make a decision. But then there's number 2. And I thought there has to be a way that I can overcome this as someone in the field just wanting to connect these 2 reserves across the road. But what this is actually referring to is the need for genetic testing. So as someone that works for council, that I once, when I started, asked for an $8 bag of sand and got told to go take a bucket to the beach. I know that genetic testing and trying to push for the funding for that is just not going to happen. Could happen, but just for the sake of this process at this point in time is not going to happen. But I can't give up yet. So I know that the writing's on the wall. I've got fragmentation. I'm seeing the low population numbers. I'm seeing the kind of imbalances in natural recruitment, and I need to keep trying. So I'm going to keep trying. And this takes us to the state government level. What do they think about this? Are they aware of this? And yes, they are. So 2020, DECA commissioned a report called the Genetic Risk Index for Victorian species. And that was under the emphasis that maintaining genetic diversity is a global conservation priority. It's written into almost all of our biodiversity-based planning. And it also makes a really good point that timely intervention, when we're considering just the whole plethora of species, not just the threatened ones, timely intervention prevents many species from reaching that threatened species status. And so the research that the report covers focuses 2 main areas. One is the genetically effective population size. So think back to those frogs. Large population, small population, what size do we need in order for these populations to be maintaining themselves and keeping the balance? And then also connectivity status between populations. So you reiterated all of my concerns. But another little chink in this kind of chain is that what they found was that only 13% of Victorian species were actually able to be assessed. So of around, I think it was around 3,600, and it's estimated that the number of species that we have in Victoria is around 4,000. So of the 13% that they assessed, only 147 of them had population structured data informative enough to create a risk index for them. So meaning that less than 6% of our Victorian species have sufficient genetic data to inform their current and long-term risk. And I thought at this point that I just must be just gone down a completely wrong rabbit hole. I must just be thinking too hard about this. But then they also incorporated the Frankham decision tree, and I thought, okay, that's all kind of connected. But it agrees, the genetic risk index agrees that testing is an important step before translocation, despite mentioning that down the bottom there that there's this ongoing decline in the feasibility of testing. So it does, I haven't put it on the screen, but the report does attempt to provide a kind of solution for this, and I'd really recommend reading it. It's very easy to read. It has a lot of a lot of graphs and that kind of thing that help you visually kind of digest that information. So it says that we can mitigate the risk when we can't have genetic testing through an evaluation of the attribute data that there is a lot of, such as breeding systems, indicative population size, the time of isolation, the environmental similarity, and that kind of thing. But for me, it uses words like relatively, risk-free. It also says we can assess the risk using those attributes to some extent, which, as I've kind of shown with trying to overcome or trying to work within some of those perspectives, that's a level of uncertainty that kind of doesn't fly in my circumstance. So kind of coming to around about the end here, and it's saying, where does it leave me? It's not quite right back where I started. It's showed me, this whole process has shown me and given me a deeper understanding of why provenance and conservation genetics has been a topic that has kind of been going around in circles for a very long time now. And it's what I've kind of come to now is that the challenge within conservation genetics isn't that we lack the recognition, don't lack the willingness to act. It's that it's such a complex system that it's resisting those simple solutions. So when we think of it like a system, it's made up of all these interdependent parts. You know, we've got ecology, we've got genetics, but we've also got policy, history, human perspectives, decision making, and each part is influencing each other in a non-linear way. So it's intersecting the past, the present, and the future. And when I came to this point, I discovered something called the wicked problem theory or concept. And it's a concept developed in the 70s by two policy and planning scientists called Rylee and Weber. And like I said at the start, I'm kind of starting to lose my voice a little bit. So I was going to discuss this in more detail, but just in the keeping things short today, if you're interested in just the amazing kind of way of looking at this problem as a wicked problem, I'd really recommend you look into that, or maybe we can go over it in another time. But wicked problems basically suggest that in order to navigate them is where systems thinking becomes really important, because when we think of things as interdependent, it gives us a way to see those relationships. We begin to understand all of these different connections, begin to see the underlying views and values that may be affecting how different management is being taken, different ways the problem is being seen. So we're shifting from wanting one right answer that solves one problem forever, because we know it's not just one problem, and we know that nothing is forever. And it's shifting that to being comfortable with bringing in lots of different perspectives and enabling us to navigate the system together And we, when we use this way of thinking, we move forward, at least from what I see, is that we can move forward with greater clarity, and that creates collective power. So I'm going to wrap up now, but I'll just make the important point that the circumstances that I described today are entirely unique, and that you all have your own unique circumstances and considerations, considering the vast and wonderful spectrum of all the different reserves and plants and ecological communities that we all work with. So I'm going to do what you're kind of not meant to do when you give a presentation like this, and it's to leave it open-ended, because I think the power of this space and synergy is that everyone coming together to use their collective experience is, you can't go it alone, these problems. Like I said at the start, I'm only one employee at the nursery, and this kind of thing is what keeps me going. So I'm going to throw it back to you as to what you think about what I've described today. What do you think is the way forward? And also bringing in your own kind of context and realising how unique that is as to what you think should be focused on. So thank you for listening, and go to, we can either break for morning tea now, or we can go on to questions, but yeah.
Do we also have to consider the insect populations, the pollination vectors that exist?
100%, yeah. So
because they're all in these different spots, and they also represent a little population that doesn't bridge anymore.
Absolutely, absolutely. If wicked problem framing, there's kind of 10 main characteristics of it, but one of them, it says that on the one hand, it can be a level of complexity that's a little bit hard to handle, but that there's no limit to the number of solutions to the problem. So that's, oh crap, like we could do everything. But also, if you wanted to focus on insect connectivity, that's a solution. So that's bringing in together, saying that we're not just going on one track to try and solve this. We've got many different ways that we can look at it and bring in that kind of thing. Yeah.
I have a question, and this is another variable which I don't know whether scientists are actually looking into, but that is pollinators. So that pollinators such as birds, flying fox and bees actually travel long distances, which probably accounts for your slide about eucalyptus species in particular being very similar when genetic testing is actually conducted, even though they're up to, 500 kilometres
yes,
from each
so,
other.
I'll bring
In the literature, I've actually read that if parks are fragmented, but they actually have water sites, and they actually have what do you call it, very large remnant trees, birds in particular will travel from bush part to another one, to another one, to another one, as long as there's a water source and a roosting tree. Possibly bats and flying foxes as well.
This presentation is a bit of a mix of different ones I've put together for my team, and usually the first slide that I have is a big photo of a flying fox, because it is that within my team there are many different kind of interests and passions and that kind of thing. Some people like myself, I really love flora, and some people are very much into conserving fauna. And again, like your question about invertebrates, is that when we start to bring in all of those collective understandings of the problem, we start to kind of gain traction as to solving it. And maybe it's not really about solving it, but we're starting to navigate it, which is, yeah.
So I would suggest to councils that they actually, if they want to increase their the problem of the fragmented parks, that they put in water sources or bog areas in all of these fragmented parks.
Yeah, yeah, definitely a very good point. And there is, I think, well, within my council, at the Grange Reserve, they're starting to look into a permanent water source. We have a lot of those little, the kind of bird bath style water that we put out in really hot weather, but they're starting to look into installing a permanent source that kind of caters across the year. And that's a very adaptive kind of way of going about it, because it's an intervention, and you know, historically it's that you don't want to intervene in that scenario, because you might be affecting these moving parts in the system in irreversible ways. But we're starting to kind of work through that, and then that's an example of it, you know, installing that water source. Yeah.
Surely everything that happens all around us is intervening in major ways. Putting something like a pond in is probably restoring the original scenario.
I know. How deep do you go when you think about it? I've gone pretty deep. Hey.
There's a lot of discussion around eDNA and being used in all sorts of fields, like even tracking of groundwater migration and all sorts of things, and it's becoming really cheap. Is this going to open up opportunities to really understand?
Well, I'm so glad you mentioned that, because I too think about things like eDNA and the kind of trajectory of a lot of these technologies with genetic testing, eDNA, and I think of just a really, you know, simple example is solar panels and electric vehicles and that kind of thing, where you know, even when I was starting uni, we would be doing projects that are like, let's encourage use of electric vehicles. But now, you know, 8 years down the track, it's almost every, you know, fifth or sixth car on the road is an electric vehicle, which we couldn't conceive back then. So absolutely, I think the key in that, though, is that we have to push for it. There has to be a need. And if we kind of are going around in circles wondering, you know, we're having all these different kind of ways of seeing the problem and that kind of thing, that it kind of limits our ability to come together and then have a collective push. But this is what this is all about today. So yes, I want to push for more funding and research and make those things accessible, and also make them accessible beyond just institutional use. So make them accessible to me on the ground. And there is, I see in the future, a way that that's going to happen. Yeah. Any other questions?
Some of this work will have to be done species by species, and how general can the conclusions be from one species?
Well, that's kind of the question that I'm trying to tackle today, is that, you know, going down all these different levels from a kind of general perspective, local providencing, been around for a while, introducing new research, introducing new technology, we get down to another level. Then we've got the guidelines that are developed. We've got, you know, these kind of tables that they present to us, and they say, you know, we've got 70 authors, we've peer-reviewed, and then the limitation of, well, I can't actually transfer Eucalyptus ovata population dynamics to another species. And that's where that kind of push, like we were just saying, for the testing, and it's, you know, I think you could, it's not really about thinking at the very start, we're going to test every single species. It's, again, working within our context as to what we see we have the most power over. And for me, it's about, you know, noticing the declines in certain species, but not necessarily focusing on threatened and endangered species, which is, I don't mean to say that that's not integral, but also I really want to prevent species from becoming threatened. So I hope that kind of addresses that a little bit.
You've presented a lot of great questions. Is there a way of contacting you to discuss further with these things?
Oh yeah, absolutely. I could chat about this for hours, and there's like other ways that I was going to talk about this. So I have interests at the moment in hybridization. So we have a species at the on the foreshore, Carpobrodus rossii, which you may have seen before, the pig face, the native pig face. And then we've got some South African species, the angled pig face and the edulus, which is the yellow one, starting to come in. And then when we do the, when we transfer the information that's been done in South Australia, for example, it says that, you know, a Carpobrodus edulus occurring within 10 kilometres is going to affect the, is going to hybridize. And then so I'm seeing that and I'm going, hang on, if it's going to hybridize and I've only got 20 kilometres of foreshore that I'm trying to manage with the species, what does that mean? So I can talk about all different things. You want to chat? Yes. I can share my email and that kind of thing if you want.
What impact does the sort of the native plant industry in terms of nursery plants have, you know, when people plant these bushes and trees in their gardens near your reserves?
Yes.
Because I remember talking to Raf and she said, oh, the correas are very difficult because they hybridise very prolifically.
Such a good question. And also a really good point of power. Where, you know, with Kingston there's so many different departments and they all have different ways that they're approaching what they need to achieve. So for example, with our horticultural department, they would be planting cultivars of Carpobrodus. And I am trying my darndest to kind of, I see that that's a really key point of power where if we can change what they're planting in that regard, then we could make a really big difference to the Indigenous populations. And also that that department, for example, is an ally in this. They have the transferable passions. They love plants, they love horticulture, they love the cultivation aspect, and to try and get that inroads into the similarities and then expand that into solutions. So I've done recently a couple workshops with our horticultural department, and you know, expanding that this is, you know, an Indigenous species, and this is how it's different from this cultivar that you're planting. But it's been good having the experience at Ball, for example, because you kind of get that that background, that insight. You know, I can think of it as like the dark side of like horticulture, and look, it kind of is in some regards, but it's it gives you that like well, it gave me that experience to kind of go, actually, this is the scale of what's being cloned, and if you were to plant this species instead, you could still achieve your the actions that you need to achieve. It's just shifting that mindset slightly. Yeah.
So all the cultivars probably come from a very limited genetic stock. You know.
They're all
I love that bush. Let's cut.
they're all clones. Yeah, cultivars are all clones.
And plant that through the neighbourhoods.
Yep.
No diversity.
Yep. Another big thing that's taking off at the moment is nature strip planting. So that has started in an awesome place where it's like, let's remove this non-functional grass and replace it with diversity, help encourage pollinators and that kind of thing. But then we're also starting to notice that we can have these guidelines that we put out that are managed by local laws and that kind of thing, but whether or not they actually have the capacity to be tackling invasive species planting and that kind of thing is another dilemma. And for example, Carpobrodus edulus is a big yellow flower, and I've gone through different websites and it's sold as native. There's been one particular website that I've tried to email a couple of times, and they've actually completely ignored me, and they signed me up to their mailing list, and then they said, if you unsubscribe from our mailing list, that blocks you from all replies to your complaint. It is. I have reported it. But yeah, so yeah, we're working with lots of different levels of information here, and yeah, it takes a lot of persistence to try and get through to the other side of some of these problems. Anyone else? Thank you all for listening. Really appreciate it, and I'm hoping it wasn't too confusing or anything like that. But yeah, thanks.